So good morning. Today we'll discuss about that um, urine formation. Urine formation. In our previous videos, already we seen that the structure of kidneys and the internal structure of kidneys and what are the functional unit of kidneys and structure of nephron and also um, filtration unit, that is the structure of glomerulus. Next, related to that, now we will see that urine formation urine formation. So nearly per a day our kidneys filters nearly 180 liters of blood will filtered by our kidneys. Among that, and then those are the 125 ml per minute, 125 ml per minute. Among that, we will pass through the urine, that is the only 1 to 1.5 liters, 1 to 1.5 liters. Remaining all that reabsorbed, 90% of filtrate, 99% of filtrate are reabsorbed again, sent into our body fluid, sent into our body fluid. So see, this is the okay, 99 reabsorbs. So the formation of urine process has three stages. One is the filtration, ultra fil, ultra filtration. Next one is ultra filtration. Next one is reabsorption, selective reabsorption, selective reabsorption. Next one is the secretion. These three process in our body urine formation has by the three these three process: filtration, absorption, and secretion. Nearly per a day 180 liters of our blood filters. So among that, only we we will mixture and we will pass through the urine only 1 to 1.5 liters. Remaining 99% of that filtrate again send into our blood or body fluids. Okay, that how urine formation will takes place. One is the filtration, ultra filtration, or glomerulus filtration. So the blood which passes through the blood capillaries in the Bowman's capsule, the the blood, the hydrostatic hydrostatic pressure of blood in the glomerulus is 60 millimercury. These pressure, these hydrostatic pressure opposed by the glomerulus glomerulus colloidal colloidal pressure osmotic pressure, that is the 32 mmHg. And Bowman's capsule Bowman's capsule hydrostatic pressure is 18 mmHg. అంటే filtration, how the filtration will takes place in the Bowman's capsule. Already in the structure of filtration unit, already we discussed that the Bowman's capsules accommodate the bunch of blood capillaries, astronomizing uh, astronomizing units of blood capillaries, that is the called glomerulus. that glomerulus causes the filtration of blood. That here to filter some pressure that needed. That pressure is how the pressure will uh, exerts by first one is the hydrostatic pressure of blood which flows in the blood capillaries in the Bowman's capsules, that is the 60 mm of Hg. Next, glomerulus colloidal osmotic pressure is the 32 mm Hg. Next, Bowman's capsules osmotic pressure 82 millimercury. These two are opposed to the hydrostatic pressure of blood, that is the 60 38 plus 32 plus 18. Net net pressure in the glomerulus is 10 millimeter millimercury. This causes to the filter the blood. After the filtration, that filtered through the three layers. That filters pass on three layers, um, that is the endothelial cells of blood capillaries, basement membrane, and those are the Bowman's capsules which has the podocytes. By the three layers of these cells, the blood filters by this net pressure, filtration pressure. The filter passes through the um, PCT area. That is the PCT. This is Bowman's capsules. The filtrate reaches the PCT. This is the proximal convoluted tube. That is called primary urine. Primary urine. First of all, so the ultrafiltration means the hydrostatic pressure of blood which flows through the Bowman's capsule. Next one that is the 60 millimercury. These pressure is opposed by the osmotic pressure of glomerulus and hydrostatic pressure of Bowman's capsules. Net filtration pressure is 10 millimercury. Because of this, by the three layers that filtrate reaches to the PCT area. This the filtrate which present here, that is called primary urine. The primary urine uh, constituents constituents of the primary urine plasma same as plasma except proteins and blood cells. So all those essential objects which are present in that filtrate again has to send into the uh, bloodstream or intestinal fluids. Okay, that by the reabsorption, that process will takes place by the selective reabsorption. So these way the real depending upon the substances of their importance, those substances are divided into three. Those are the high threshold substances. High threshold substances, low threshold substances, a threshold substances, a threshold substances అంటే which are depending upon that we will know that we will need that what are the substances has to reabsorb necessarily. Okay, high threshold substances means they have to compulsory by the active or transport, by actively or passively they have to reabsorb by the renal tube. Those are the glucose. Next, amino acids. Next one is glucose, amino acids, water, and some electrolytes. These are high threshold substances. Next, low threshold substances. These are the urea and uric acid. A threshold substance is creatinine. This is the excretory product. Why? That's why there is no need to reabsorb, that is the creatinine. So see these by the selective reabsorption or absorption process, the in the PCT area, in PCT area in PCT area, uh, 80 80 to 85% of filtrate, 80 to 85% of filtrate again reabsorbed by the PCT area. This PCT area has the cuboidal brush border epithelial cells are present. Cuboidal brush border epithelial cells are present in the area of PCT. This PCT area is the main process is selective reabsorption. Selective reabsorption, 80%, 80 to 80% of 80 to 80%, 85% of electrolytes and water and water reabsorbs and send into the bloodstream. Next one, so here next one, secretion. So some hydrogen ions, potassium ions, also next ammonium ions, all these are secreted, these are the secrete, these are the secrete by the cells of PCT area. Okay, so next, the charged ion of sodium, uh, positive ion sodium ion actively transported into the bloodstream. This helps to move the driven to the chloride ions. Here the PCT area, the primary urine which is present in the PCT area, these constituents are nearly equal to plasma. That's why all essential objects which are present in the primary urine has to send has to has to absorb and sent into the bloodstream by the selective reabsorption. Selective reabsorption, those are the glucose, amino acids and uh, water, some electrolytes are reabsorbed and sent into the bloodstreams. Along with secretion also participate here to maintain the ionic balance, osmoregulation regulation and acidic base acidic base balance. It is a necessary, it is also main important to secrete these hydrogen ions, uh, potassium ions and ammonia. Okay, these Na plus positively charged ions actively transported to the bloodstreams. This helps to driven the chloride ions, negatively chloride ions also to move. Okay, next one is the, this is the secretion and absorption. Next one is this the descending loop. descending Descending with the glomerulus filtration. The primary area is hypotonic in nature, hypotonic solution. Next one, the descending loop. In the see here, descending, this is a descending loop, this is a ascending loop. This descending loop uh, will not permeable to the salt solutes, only it is permeable to the water, and the water which is present here sent to the lumen, sent to the interstitial fluid, and also not for the solutes, not for the electrolytes. This makes the filtrate will make high osmotic concentration. When it moves upward ascending limb, this ascending limb allows the, it is not permeable for water, only for the salts, and the salts which is the sodium, calcium, potassium, all these are reabsorbed here, not for water. And when this solution when this solution moves upward to the cortex, that makes the interstitial fluid dilute. Dilute means hypertonic. It makes the hypotonic This makes the sorry, this makes the hypertonic. This makes the hypertonic. Next one is the TCT. DCT, distal convoluted tube. Distal convoluted tube. This distal convoluted tube, here the cells are short very short. That's why these are not participate actively in the reabsorption, but in some special conditions, by the influence of ADH, anti-diuretic hormone, it reabsorb reabsorbs the water and some, these are the glucose amino acids, along with they secretes these uh, hydrogen ions, potassium ions, and ammonia. Next one, all the remaining high concentrated urine sent into the collecting duct. In the collecting duct, so by the ADH or vasopressin, the remaining water which is excess, that is reabsorption. After that, the concentration urine will send into the urinary bladder through the urethra. This is the urine formation process. Nearly per a day, our kidneys filters the 180 liters of blood. Among that, we will pass out only the pass the urine only 1 to 1.5 liters of the blood, and the remaining 99% of filtrate, 99% of filtrate will reabsorb and send into the blood capillaries or interstitial fluid. This urine formation process has three stages. One is the ultrafiltration or glomerulus filtration. Next one is the selective reabsorption. Next one is the secretion, active secretion. Okay, first one is the ultrafiltration or glomerulus filtration. That the net pressure of 10 millimeter mercury will causes the filtrate. Those are the uh, uh, after filtration, the primary urine reaches the PCT. In the PCT area, maximum amount of 80 to 80% of 85% filtration will reabsorb here. After reabsorption, the filtrate which moves towards the descending loop. This descending loop permeable to the water, not permeable to the electrolytes. That's why this urine will concentrate. They will concentrate and makes the interstitial fluid into hypo hypo hypotonic. Okay, after that, the this this urine will moves towards the upward, towards the cortex. This thin loop of this ascending limb allows only electrolytes, not permeable to the water, not permeable. Then the urine will concentrate, and this uh, medullary fluid and cortical fluid may dilute after, when that reaches the DCT area. In the DCT area, special cells, chief cells are present, so they are influenced and works by the influence of ADH, anti-diuretic hormone, and some amount of remaining, these all are reabsorbed and sent into the collecting duct. From the collecting duct, the concentrated urine will send into the urinary bladder. Whatever that urine, so when we take lot amount of water, we may more amount of more amount more value more amount more volume of urine will pass out, whatever that volume of water. But here solutes, waste materials are constant, that is the 600 milliosmol per liter. These are the waste materials. These are the waste substances which are present in the urine. Whatever, when we are in deprived to water, మనకు వాటర్ అందుబాటులో లేకపోయినప్పుడు, వాటర్ తాగనప్పుడు, the urine, less volume of urine will pass out. In that less volume of urine, and when you take more amount of water, larger amount of volume we pass out. What There is no uh, change of waste materials in that urine. That is the constant 600 milliosmol per liter. Okay, this is about that urine formation. In next class we will discuss about that concentration of urine, the the urine which is passes through the urinary tubules, how it is concentrated. Okay, thank you.